Good evening. Myself, Dr. Anu Sharma, Assistant Professor, Beta Technical University, Bikaner. Today I am discussing the topic structure of protein. Protein are the macromolecules and they are most abundant organic molecule in the living system. They consist about 50% of cellular dry weight. They constitute the fundamental basis of structure and function of life. They are the polymer of amino acid. Protein derived from the Greek word that is proteos. It means first place. Protein are the nitrogenous macromolecule that are composed of many amino acids. Now we will discuss the amino acid. Amino acids are the group of organic compound containing two functional group amino and carboxyl. This is shown in this figure. In right hand side there is carboxyl group and in left hand side there is amino group. The amino group is basic in nature whereas the carboxyl group is acidic in nature. There are about 300 amino acids occur in nature, only 20 of them occur in protein. Now the structure of amino acid. Each amino acid has 4 different groups attached to alpha carbon. Alpha carbon is the adjacent to carboxylic group. It has amino group that is NH2. It has hydrogen atom H and alkyl group R and COH group carboxyl group. These are few examples of amino acids. In the first box, these are non-polar aliphatic R group. Example is glycine, alanine, in which R group is non-polar. In the second box, right hand side, these are positively charged R groups. For example, lysine and arginine. We are saying positive charge on NH2 group. In the third box, in lower left side, these are polar uncharged R groups. Serine, for example, CH2OH in serine. This is polar uncharged R group. In the right hand side, middle box, these are positively charged R group. And in lower portion, aromatic R group. For example, phenylalanine, tyrosine. Peptide bond formation in protein. Alpha carboxylic group of one amino acid forms a covalent peptide bond with alpha amino group of another amino acid by removal of water molecule, and it is formed dipeptide. When two amino acids combine, dipeptide bond is formed and this is CONH. This is CONH. The dipeptide can form a second peptide bond with third amino acid, means tripeptide. And repetition of this process generate a polypeptide or protein of specific amino acid structure. It has 40% double bond character caused by resonance, means peptide bond has 40% double bond character. This is the example of formation of peptide bond. Amino acid 1 in which carboxyl group, amino acid 2 in which amido group, they combine. There is elimination of water molecule and in peptide bond form CONH in the lower structure. Polypeptide backbone is the repeating sequence of NCC. Here we see the backbone NCC, NCC means NH2CHR1CO, then again NHCHR2CO and so on. The side chain of R group is not part of backbone or the peptide bond. Chain is shown in lower part R1, R2, R3 and R4. Overview of the protein structure. The key feature of protein structure are configuration, geometric relationship between a given set of atoms, conformation, spatial arrangement of atom in a protein. Thermodynamically, most stable conformations exist and stabilize largely by weak interactions. Stability arises due to maintained native conformation. Native conformation stabilized by disulfide bond covalent forces, non-covalent forces. The structural organization of protein have four type of structural organization, that is primary structure, secondary structure, tertiary structure and quaternary structure. The key force of primary structure is determined by covalent bond, whereas in secondary, tertiary and quaternary structure determined by the weak forces. Let us discuss the overview of different type of structure. Primary structure means assembly, secondary structure means folding, tertiary structure means packing and the quaternary structure means interaction. Now we will discuss the primary structure. The primary structure of protein refers to the sequence of amino acid present in polypeptide chain. These are covalently bonded and the bond is known as peptide bond. Each component amino acid in a polypeptide is called residue or moiety. And in primary structure, the bond starts from amino terminal end and ends in the carbonyl terminal end. The importance of primary structure. These give the structural prediction of secondary and tertiary structure of protein. Many genetic diseases result from the abnormal amino acid sequences. It gives the molecular mechanism of action of protein and it traces the evolutionary path. This structure can be determined by using end group analysis, that is Edman degradation, and the gene sequencing method. Now we will discuss the secondary structure. Secondary structure has localized arrangement of adjacent amino acid formed as polypeptide chain forms. It consists alpha helix, beta plated sheet, beta bands, non-repetitive structures, super secondary structures. Few arrangement are shown in the upcoming slides. Alpha helix. It has spiral structure. It is tightly packed, coiled polypeptide backbone core. Side chain extend outwards. We are seeing R in green outwards. It is stabilized by hydrogen bond. We are showing this by arrow, hydrogen bond. Hydrogen bond between carbonyl oxygen and amide hydrogen. And amino acid per turn are 3.6. Its pitch is 5.4 angstrom. And examples, globular protein like myoglobin, troponins. There are many types of helix. Alpha helix, as we shown in the previous slide, it is also known as 3.612. Pi helix, very loosely coiled hydrogen bonding pattern. 310 helix, very tightly coiled hydrogen bonding pattern. These all three are shown in this figure. Now we will discuss the beta plated sheet, formed when two or more polypeptides line up side by side as shown in the figure. Individual polypeptide beta strand and each beta strand is fully extended as shown in this figure. They are also stabilized by hydrogen bond. There are two types of beta sheets, anti-parallel beta sheets and parallel beta sheets. In anti-parallel beta sheets, neighboring hydrogen bonds, polypeptide chains run in opposite direction. It is shown in figure A. Arrow are in opposite direction. Whereas in parallel beta sheet, hydrogen bond chain extended in the same direction. In the B diagram, all arrows are on same sides. The connection between two anti-parallel strands may be just a small loop as shown in A figure, but the link between tandem parallel strands must be a crossover connection that is out of the plane of the beta sheet as shown in the B. It is out of the plane. We can also define these connection like a hairpin or same end connection and shown in as it is shown in figure A, hairpin structure, and the crossover or opposite end connection as shown in figure B and C, in which they are crossover or opposite end connection out of plane as we discussed in previous slide. The angle between two intersecting planes is known as dihedral angle or torsion angle. It helps to maintain the protein structure. It is very important to maintain the protein structure. Now, protein foldings. The peptide bond allows for rotation around it, and therefore the protein can fold and orient the R group in favorable position. Protein folding occur in cytosol, as shown in this diagram. Two regular folding patterns has been identified formed between the bonds of peptide backbone alpha helix in figure 1 upper side and beta sheet is shown in the figure lower side. There are many turns and loops are also present in these structures. Loops and turns connect alpha helix or beta strands. The most common type cause a change in direction of the polypeptide chain, allowing it to fold back on itself or create a more compact structure. Loops have hydrophilic residue and they are found on the surface of the protein. Loops that have only 4 to 5 amino acid residue are called turns when they have internal hydrogen bond. Reverse turns are a form of right turn, whereas the polypeptide chain makes a 180-degree change in direction. There are beta turns are also present. These are also known as beta bands or tight turns. In a beta turn, a tight loop is formed when the carbonyl oxygen of one residue form a hydrogen bond with the amide proton of the amino acid 3 residue down the chain. This hydrogen bond stabilizes the beta band structure. A beta turn means by which the protein can reverse the direction of its peptide chain. Beta turns often promote the formation of anti-parallel beta sheets. This diagram shows the secondary structure. Now we will discuss the tertiary structure. Tertiary structure is the three-dimensional conformation of a polypeptide. The common feature or protein tertiary structure reveals much about the biological function of the proteins and their evolutionary origins. Tertiary structure is shown in the figure. The function of protein depends on the tertiary structure. If it is disrupted, it loses its activity. Tertiary structure is stabilized by several bonds, that is, hydrogen bond, ionic bonds, disulfide bridges, and hydrophobic interactions. In the figure, right hand side, there is hydrogen bond, hydrophobic interaction, van der Waals interaction in pink, in yellow, disulfide bridge, and in lower blue, ionic bond is formed. So these bonds stabilize our tertiary structure. For example, in globular protein, tertiary interactions are frequently stabilized by sequestration of hydrophobic amino acid residue in the protein core. Consequent enrichment of charge or hydrophilic residue on the protein's water exposed surface. In secreted protein, disulfide bond between cysteine residue helps to maintain the protein tertiary structure. These are the example of tertiary structure disulfide bond. There are two cysteine amino acids. Between these, a disulfide bond is formed, as shown in this diagram. Hydrogen bond also stabilize the tertiary structure. Hydrobonds are the weak which allow to be broken and reformed easily. It is shown in this figure. The hydrogen bond formed between carbonyl group and amido group. Salt bridge also stabilize the tertiary structure. These are electrostatic bonds between oppositely charged groups. The strength of this bond is 4 to 7 kilocalorie per mole. Ions of R group from salt bridge through ionic bond. It is shown in this diagram. Hydrophobic forces in tertiary structure. Close attraction of non-polar R groups through dispersion forces, as it is shown in this shaded structure. They are non-iterative interactions but results from the inability of water to form hydrogen bond with certain side chain. Very weak but collectively interaction over large area of stabilized structure we are seeing in this figure. In brief, we can say hydrophobic interaction The nature of it, this type of bond, attraction between nonpolar alkyl and aromatic group from a nonpolar center that is repelled by water. That is, example, methyl means alkyl group. Second, hydrophilic interaction, attraction between polar or ionized R group and water on the surface of the tertiary structure. In second, CH2OH and water. Salt bridge, ionic interaction between ionized R group or acidic or basic amino acid is shown in this figure, carboxyl group and amido group. Hydrogen bond occur between polar side of group of amino acid. And last, disulfide bond, strong covalent link between sulfur atom of two cysteine amino acid, as we discussed it in previous slide. This is the picture of tertiary structure. This structure can be determined by X-ray crystallographic studies and nuclear magnetic resonance studies. Now, the quaternary structure. The biological function of some molecule is determined by multiple polypeptide chain, as shown in this figure. These are multimeric protein. Example is hemoglobin, which is found in our blood. Quaternary structure of protein involves the clustering of several individual polypeptide or protein chain into the final specific shape. A variety of bonding interaction including hydrogen bonding, salt bridge, disulfide bond hold the various chain in a particular geometry. These are of two types, homodimer and heterodimer. Homodimer means association between identical polypeptide chain, whereas heterodimer interaction between subunit of very different structure. The interaction within multiple subunits are the same as that found in tertiary and secondary structures. Quaternary structure adds stability by decreasing the surface by volume ratio of smaller units. Simplify the construction of large complex viral capsids and proteasomes. These are the figure shown the quaternary structure of protein. This is the example. There are two tertiary structure in blue and green. When they combine, they form quaternary structure. Right hand side. Here, in brief, we can say in left side, secondary structure in which contain alpha helix and beta sheet. Tertiary structure in middle portion and quaternary structure when two tertiary structure molecules are combined. They are same protein structure or different protein. In summarize, primary, the sequence of amino acid. Secondary structure, the coiled alpha helix, beta pleated sheet or the triple helix formed by hydrogen bonding between peptide along chain. Tertiary, a folding of protein into compact three-dimensional shapes stabilized by interaction between side R group of amino acids. And the quaternary, a combination of two or more protein subunits to form a larger biological active protein, for example, hemoglobin. Now, we are facing the pandemic situation, and the main cause of pandemic situation is COVID. The structure of COVID contains a protein. Its envelope protein, membrane protein, and spike protein. Outer we are shown in this figure. Below this, nucleocapsid protein and a lipid membrane is also found. So this protein also form a COVID structure. Thank you very much.